I would like to add my voice to thanking Aniridia Sweden and Aniridia Europe and Neil for the kind invitation to talk here today. As Peter has shared with you already, I'm part of Aniridia North America, and it's a pleasure to be representing both the research that I do in Aniridia North America. What I would like to do is to talk a little bit about the non-visual parts of Aniridia. So many individuals with aniridia report symptoms that are in addition to vision. So a loss of a sense of smell or a mild impairment in the sense of smell, trouble hearing speech or spoken word in a noisy environment, trouble understanding speech content, again, in a noisy environment usually, increased sensitivity to touch and pain, and trouble with the sleep-wake cycles. In addition, aniridia has been associated in the literature with ptosis, cerebellar ataxia, and altered cognition. So the question becomes, do people with aniridia really experience more of these neurological symptoms than do people who don't have aniridia? And then the second question that immediately has to follow that is, can these symptoms be explained by a reduced PAC6 activity? And so what I think is that we have a significant gap in our knowledge currently. And that gap is really, what is the prevalence of neurological symptoms in people with aniridia? So as a beginning point to answer this question, Janelle Collins in Aniridia North America had initiated a patient-driven survey. And so this is the first report of these results. This is very new data. We just simply, this survey was done in January of this year. And what I'm showing you is a very conservative analysis. We are still processing this data, but I feel comfortable sharing at least a couple of these with you. The first is that if we ask, what is the prevalent, and so what I'm showing you are individuals who have a clinical diagnosis. So the number of individuals is the number of individuals in the survey. The number of people who are reported, 16, 19, and so forth, are those who actually have a clinical diagnosis, and so we're not making any guesses here. And what you can see is that about 10% of the patients, individuals, are clinically diagnosed with an auditory processing disorder. If, for comparison, in the United States population, this symptom usually comes at about 1%. If we look at sensory integration processing disorders, we see 11%. This number has a wide range depending on who is doing the diagnosis or how the diagnosis is being done. And so this captures a lot of different things. Our own data suggests that we're closer to 5% if we take out autism spectrum disorder. For individuals with aniridia, about 10% are clinically diagnosed with autism spectrum disorder. And then I was surprised to see that actually 8% were diagnosed with obsessive compulsive disorder, compared to about 1 to 2% in the general population. So it appears that there are, in fact, some clinically diagnosed changes in neural processing. If we ask about motor control and vestibular control, so this data is probably the most raw at the moment, but the results are pretty clear. So out of 196 individuals answered or that gave us information, 39% reported motor control issues. Now, this can be broken out into many different ways. The two that I'd like to bring your attention to are gait problems, and because I know this question is coming up later, epilepsy. And I'm happy to talk more about the epilepsy data when this topic comes up during general discussion. In terms of vestibular, we see that 83 of 196 people, or 43%, have reported balance problems. Now, this one's tricky because we haven't factored in age differences. And as an older person, I can tell you that my balance is a lot less than it was when I was younger. My daughters will tell you the same thing. So we have to be careful with this. And so it seems clear that there are changes that we need to pay attention to. So the next question is, where do these changes come from? And so it can come in one of two ways. So we could see altered tissue development. And so that's captured sort of in a simple cartoon over here. Let's say that PAX6 is required to assemble these cells in this order. If there is, it may be that a reduction in PAX6 causes that program to misfire. And so then you get a different set of cells in the tissue, and the tissue has to alter what it does. The other way this can play out is in an adult. And now when I say adult, I mean anybody after birth is considered an adult to a developmental biologist, okay? So we're not talking about adults here. So there could be altered cellular physiology in the adult tissue. I've depicted that by cartoon. So in maybe PAX6 is required to put in 4 blue bars between the different cells. And because PAX6 is reduced in people with aniridia, by 50%, they only have half as many of those blue bars holding those cells together, right? And that would, in this kind of context, would make for a more fragile epithelium, for example. So now I apologize, I have to go, because I'm a molecular biologist, I have to do this. So PAX6 is a transcription factor. That means it's a protein that's controlling many other genes, especially other transcription factors. And so it's sort of diagrammed in this very lovely cartoon. So PAX6, the protein, is sitting down on DNA, and it can either turn on gene expression. So example, if you see the green arrows, so PAX6 is turning those genes on, allowing them to be expressed. But it can do the opposite too. It can actively suppress expression, and that's captured over here. Now, the thing about the PAX6 protein is it's a very complicated transcription factor, and I'm using that in a professional sense. It can bind to DNA in many different ways, and it can activate many, many different gene networks. And so that's captured over here. Just if you're wondering, right, we can look at the DNA sequence that it actually sits down and binds. And this is not my data, this is from Oliver Sveckel's lab, but it captures the stuff that we get in my lab all the time. And so if you ask how many places does PAX6 bind in the chromatin, for the developing cortex in the mouse, we see that number is like 2,400. If we look in the developing lens in the mouse, it's about 2,300. And if we look in endocrine beta cells, right, you see a smaller number, but it's still a large number. It's about 1,000, 1,200. And that's pretty true. Whatever tissue or cell we look at, especially if it's early in its development, we see about 2,500 binding sites. That just means that the PAX6 protein has the potential to turn that gene on or off. And so now this is a complicated diagram. I tell my students never do this, and so of course I had to do it for you guys. But the point is, you don't need to look and know what's here to get the point. The point is that PAX6 sits in the middle of this network, and it can regulate many different processes in cells. So it can regulate whether a cell is a neuron or something else, or it could regulate the type of neuron. So it could be something that inhibits or a neuron that activates. And we know that that changes depending on PAX6 levels. It can also control things like metabolic regulation. Now, metabolic here means inside the cell, but we're going to get to the broader context in a second. It can regulate transcription, chromatin, cell cycle, etc. This complicated diagram over here is, in fact, from my lab. This is for Neil, mostly. Because what I've done is I've shown you a network that controls how neurons talk to each other. And everywhere you see a blue circle, those genes are directly turned on or turned off by PAX6. And the pattern that you see here that jumps out is that it's capturing the protein that turns on the pathway, and it's capturing the protein that actually listens to that other protein. And so everywhere you see a blue circle means that these two things are talking to each other, controlling everything that's on the left-hand side. And so the cells pay attention to how much PAX6 is there. And so the only point that I want to make with this is that what you're seeing in blue versus red, so we took a single cell type and we said, okay, I'm going to take away half the PAX6 and just ask what happens. And so what you're seeing by comparing these two things is, yes, in fact, there's a change in gene expression. We expected this. The important thing here is that we know what genes are changing in this data. These complicated networks are kind of interesting to look at but maybe not very helpful, except for the fact that there are a lot of signaling pathways in there that we can use to begin to manipulate how PAX is acting in a cell. And so now, back to sort of where we're going. So the first question is, are there in fact structural changes in the human brain that we can see? And the answer is yes. And so this is just a comparison of data from my lab and other labs. The important thing is that we can, yes, in fact, we can see a change in specific structures in the brains of people with aniridia. Do all people with aniridia have the same changes? The answer is no. But the changes that we do see are consistent in a pattern. And just a couple examples. So there are a couple important structures here. In this case, we have these two commissures that are bridging the brain here. So this is our individual that is PAX6 normal. The structure down here is the pineal gland. And so in individuals with aniridia, we often see a reduction in the anterior commissure, this posterior commissure, and the pineal gland. Again, it varies a little bit between people, as you would expect. And this is just a different look. So this major structure up here at the top in the red box is the main communication conduit from the left and the right sides of the brain. We can see a slight reduction in this in many people with aniridia. And then the optic chiasm, which is the receiving or sending information out of the retina, is also reduced, but that one is very understandable because PAX6 affects the retina. So a lot of the output from the fovea, specialized structure, routes through the optic chiasm. So if the fovea is reduced, it makes sense that the chiasm is also going to be reduced. Importantly, though, there are parts of the brain that are not changing. And so we didn't see any changes in the corpus callosum area. That means the whole structure is roughly normal, as far as we can tell. Gray matter, so that's the number of neurons. White matter, that's the talking between the different neurons. And total volume, brain volume. Now, there are some reports of changes, which are reports of changes in brain volume in people who are older. And I'm going to show you that data in just a second. The important thing, though, is, again, all of us are losing brain volume as we age. I can guarantee that my brain is smaller now than it was 30 years ago. Part of that's lifestyle, I'll have to add. And so PAX6 is, so we're going to talk about our main model system, which is a rodent. And so PAX6, you're looking at blue expression in a developing mouse embryo. Wherever you see blue, that's PAX6. The important thing for brain development is that, shown here on the far side, is that PAX6 is actually expressed in a graded fashion in all vertebrate brains that we know about, and presumably the same is true for human. So the structures that are predominantly affected in people with aniridia are the structures that tend to sit at the front of the brain, or the structures that usually see the highest concentrations of PAX6. So the idea is that reducing PAX6 a little bit is probably causing those cells to start misfiring a little bit. This one's a little harder. I just want to make the point, this is a mouse, but it's also true for human brains, is that we do see PAX6 expression in the adult brain. Again, you're looking for purple. This is the olfactory bulb, which is very big in a mouse. This is the cerebellum, and I want to pick on this one in a second. This is the part of the brain that processes memory. It's called the hippocampus. We also see expression in other parts of the brain down here, and I'm going to bring those into the story in just a second. This is a mouse brain. Now we're showing you a live expression just to make the point that there's actually a lot of PAX6 expressed in the mammalian brain. So we can explain the structural changes by understanding a little bit of the change in PAX6. But what is the significance, if any, of the structural changes? So to answer this question, we're going to shift over to imaging data. We're going to do this in two ways. So the first test that we're going to do is an auditory test, and we're going to measure brain activity using electrical activity. And the test that we're going to do is an involuntary response that occurs in all people, right? You cannot control this thing that we're about to do. Your brain, if we play a tone, your brain responds, and that is true for every person, and it happens every time. And so what we're going to do, so I have to reference the first studies by 2004 and 2007 that showed that people with aniridia have a deficit in interhemispheric auditory transfer. Okay, that came off the structural stuff. What we did is we used this activity, and what you're going to see is that people with aniridia, every single person we've tested, has a change in brain activity in response to this tone. And so so you can see this. This is what the response is from actually one of my students. I shouldn't tell you that, but it's true, right? And so what you're looking for is the peak here. There are 3 different tones, the 22, 40, and 84 hertz. And you can see that individuals with aniridia have a much higher response at all of these tones than a person who is PAX6 normal. Now, what does this mean? We don't really understand. It certainly is tied to auditory processing. What it means is the brain has to work harder to process certain types of information. Again, it's a little bit like an aging phenomenon, because I can tell you as an older person, having done this, that my pattern has gotten, also more robust as I've gotten older. All of the individuals in this study were in their 20s and 30s, okay? So we know that there is a functional change in processing. This is another way to look at this. This is using fMRI, so we're looking at brain activity. I have to warn you that this resolution is from somebody who usually works at the cellular level. This is terrible, but it's the best we've got. And the important thing here is that wherever you see, so what you're looking at are different parts of the brain that are color-coded. So green is under executive control. Red is primary visual, so that's simply measuring processing of visual information. And then this blue bit is what we call the intrinsic default mode network. This is the network that is not stimulated by sensory input. It's the part that when you're thinking about things that works. And what we've noticed in individuals with aniridia is that these areas are broader than people who are PAX6 normal. And what the implication is that brain is working harder again, or you're using more of the brain to process information. This is the study that reported the greater decline in the thickness of the frontal temporal cortex and an accelerated decline in working memory. This sounds scary, but again, a lot of the individuals in that study were older, and so what we don't know is, how much of this is just due to aging. So then, on a light touch. So PAX6 is expressed by cells outside of the brain too. And so, sorry, that was a silly statement. PAX6 is expressed by other cells, mainly the ones that are in the endocrine system, and they're going to take us into 2 places. So we know that the PAX6 is expressed in the pineal gland. I just showed you some of the MR data. The pineal regulates circadian rhythms, and it tends to be reduced in some individuals with aniridia, but not everybody. If we look over and it's slightly adjacent, so there are 2 groups of neurons. There are neurons that we call neuroendocrine neurons in the hypothalamus, and we also look at neurons in the pituitary gland. Now I'm shifting to mouse. And so we know that in our mouse model, there's a change in the cellularity of the pituitary gland. There's an increase in thyroid-stimulating hormone-producing cells, and we see increased levels of thyroid hormone in our aniridic mouse model. Now, this hormone controls many other metabolic processes. In our pilot data, we see also an increase in circulating levels of TSH-producing hormones in people with aniridia. Again, there's a lot of variability, and it's a subtle change, but it's measurable. In our neuroendocrine neurons in the hypothalamic region, we see altered levels of hormones in our PAX6 deficient cells in culture. So that tells us that PAX6 is having an impact on the way that those cells function, but we do not know what that means in the context of either a mouse or a human being. But we know that it is changed. Last but not least, PAX6 is expressed by GLP-1 expressing L cells in the small intestine. So that's PAX6. I'm not showing you the GLP-1 at the moment, but I need to have this here. It's also expressed, and this is well documented, by the endocrine cells in the pancreas. And so this is a mouse pancreas, and everywhere you see green, those are expressing PAX6. And the important thing about this relationship is that the L cells are producing this enzyme GLP-1, which is feeding information from the intestines when you're eating food over to the pancreas, and it's feeding this into two ways. And so we know that PAX6, in cultured cells, GLP-1 production is reduced, and in the pancreas we know that there's an altered production, release of insulin, and an altered function of alpha and beta cells in the pancreas. Again, subtle, but measurable. The implication is that this regulation, a shift in this regulation, can lead to an increased propensity for diabetes. And the corollary to that is that the way that this is changing is not the typical form of diabetes that you find in somebody who is has acquired it through other mechanisms. And so the main takeaways. And I apologize because you've got some salesmanship going on in here. But the first is that aniridia is more than just an eye disorder. And we know that because PAX6 is expressed in more than just the eye. The premise of everything that I'm telling you is that tissues that require PAX6 for normal development, and I'm using this in a genetic sense, that means if PAX6 is missing, those tissues do not form or do not form properly. So that says PAX6 is needed, may also exhibit altered development when we give it some PAX6, but not all of the PAX6 that it needs. The other way that this plays out is that cells in adult tissues that express PAX6 can exhibit altered physiology to changes under certain conditions, right? So some cells respond to reduced PAX6, some cells don't seem to worry about it all that much. So PAX6 is a sequence-specific transcription factor, and so then that means that it can control cell physiology. Some gene networks are controlled by PAX6. Some of the networks we know are dosage sensitive. That means that they are turning off or turning down, whereas other parts of the PAX6 pathway are simply unaffected. And again, that's to be expected. People with aniridia have structural and functional alterations in the nervous system. We can see this most easily in the context of vision, smell, and hearing, sleep-wake cycles. This bit with cognition is a bit tricky, and I'm happy to come back to this. We also know that people with aniridia may have changes in metabolism and endocrine function, and they do appear to be at higher risk for diabetes, and that's established in the literature. And so here's the salesmanship. Understanding the gene networks controlled by PAX6 gives us the tools, it gives us the candidates that we need to begin to structure or come up with design-driven approaches to controlling PAX6 activity. Now, we're not trying to control PAX6 itself, but we're picking the pathways that are down below PAX6 to try to tune them so that they can do their jobs better. And, right, that's a long process. But it begins with understanding who PAX6 is talking to and how it's doing it. And so with that, like Peter, I would like to thank a lot of colleagues, especially the people who participate in our studies, people with aniridia. Right, I covered a lot of territory, so it takes a lot of people. And funding, but I also want to acknowledge, in particular, a couple of funding sources that are not federal. And a lot of this research began with the Sharon Stewart Aniridia Trust. Sharon had aniridia, and she was frustrated, and so she began to fund research. And some of the ideas that were funded with the initial Sharon Stewart monies, private monies, are now beginning to yield possible clinical translational results. And so it's taken a few years, but I owe a debt of gratitude to her. And I'd also like to thank my colleagues in Aniridia Europe, because the interactions that we're having at this meeting, at the meetings that we have in the United States, are so vitally important to moving ideas back and forth, right? The chance conversations that we have here really get us going in terms of, gee, did you think about that? or to view the world from a slightly different perspective. Lastly, a lot of the research that we do is animal-driven, and so we need really good animal models. And so Beth Simpson's going to be giving a talk Sunday, I believe, with a really exquisitely and powerfully built new set of animal models for understanding aniridia. That research is absolutely critical to moving it forward, especially given where we are now. And I believe Dr. Musajay will be giving a talk on metabolomics on tomorrow. And so hopefully I've set her up well enough. And with that, thank you all for your kind attention.